everybody. Thank you for coming today. I'm Laura Colgan, the Director for the Center for Learning and Memory at the University of Texas at Austin, and a member of the SFN Program Committee. I'd like to remind you of a few housekeeping items before we get started with this exciting lecture. Please silence all of your electronic devices to minimize interruptions during the presentation. And please respect our photography and recording policy, which is that you are only allowed to photograph or record the presentation if the lecturer has a green camera with a checkmark image on their slides. In the absence of this icon, all recording or photography of any kind is prohibited. There will be a Q&A with Dr. Suthana at the end of the session, so please stick around to hear her answers to your questions. And to submit questions, please use the Q&A feature available in the session description of the meeting app or virtual platform. You may submit your questions at any time during the presentation or during the Q&A segment. If you need to leave before the end of the Q&A session, please sit near the back of the room and please leave quietly so as not to disrupt the presentation. This session is being recorded and will be available within 24 hours and accessible through the virtual platform through December 21st. And with that, it's my great pleasure to introduce Dr. Nanthia Suthana. Dr. Suthana's research lies at the cutting edge of human cognitive neuroscience and translational neuroengineering. She is a professor of neurosurgery and biomedical engineering at Duke University, and since 2015, she has served as the associate director of the neuromodulation division of the Semel Institute of Neuroscience and Human Behavior at UCLA. Dr. Suthana's unique and powerful research employs invasive neurophysiological and neuroimaging techniques, as well as deep brain stimulation, to investigate the mechanisms of learning and memory and spatial navigation in the human brain. Her work is also highly translational, suggesting novel neurostimulation-based therapies for multiple neurological and mental health disorders, including post-traumatic stress disorder and memory disorders, including Alzheimer's disease. A major innovation of her work is the use of real-world tasks and mobile intracranial recordings in humans, which allows her to elucidate how we keep track of where we are in space and how we encode and retrieve our memories as we move freely through the world. She is also a gifted communicator, as you're about to see, so you're in luck. You're going to enjoy yourself during this presentation, in addition to learning a lot. So without further ado, please join me in a warm welcome for Dr. Nanthia Suthana.
Thank you, Laura, for the kind introduction, and thank you to SFN for inviting me here today. It's a huge honor and privilege to represent my lab team and share with you what we've been up to over the last several years. So, my interest really lies in understanding how the brain supports navigation not only through physical space, but also through our memories. And it's the potentially shared mechanisms between these two that may underlie our ability to mentally time travel, including in the vivid flashbacks that you see in PTSD. By understanding how the brain organizes experiences across time and space, our hope is that we can help contribute to therapies for patients with memory disorders. All right, along the way, I might include a few little snippets of text in the top right that have to do with my personal journey and how we got here today. So let's get started. First, I'd like you to just think back to this morning. I'd like you to replay the walk from your hotel to this conference room. Picture the sights, the sounds that you saw along the way, crossing the street, grabbing coffee. This is your brain's navigation and memory system really working together. This is how it looked for me yesterday. You might recognize this walk if you're staying in the same hotel. And, this ability to essentially mentally time travel and go back into our experiences is critical for what we know of as episodic memory, or the ability to, form memories for personal experiences. For a long time, we couldn't record directly from the human brain during an experience such as this. But today I'd like to tell you how that's changing. And I've included a reference for you that we wrote recently on this topic. Now imagine trying to replay that walk and nothing comes up. This actually happened to me once. I woke up in a hospital, not remembering how I got there. Luckily, it was very temporary. But I can tell you from personal experience that that memory was just not there. It was not accessible. It did come back soon, and no amount of effort to try to bring it back, no matter how many times people say, just try harder, do you remember? I could not. It was completely blank. And this is the reality for a patient that we all know of probably, Henry Molaison, otherwise known as HM. Henry suffered from epilepsy at the early age of 9 years old, and he underwent a surgery to remove his bilateral medial temporal lobes, as you can see here in red. It was through the work of Brenda Milner, a neuropsychologist who worked with Henry and helped us understand that this region, essentially a region within this region called the hippocampus, was critical for our ability to form memories for our personal experiences. Now, I heard about the story of HM and Brenda's work when I was an undergrad. I actually dropped out of school, so let's call me an undeclared undergrad. I was uninspired, and I heard of Henry's story, and it really sparked a curiosity, rather I should say an obsession, with understanding how this brain region could hold the key for our ability to essentially just remember events in our life, something that we take completely for granted and we do every day. But when lost, completely devastates a person's, quality of life. Now, I went back to school and I majored in neuroscience at UCLA, and it was there that I learned about the pioneering discoveries of John O'Keefe and the Moser labs who won Nobel Prizes for discovering that cells in these regions actually respond when an animal is moving through space. Here you can see many different place cells shown in different colors that increase in firing when the animal is in a certain location. And together, they form a sort of neural map of space. Now, these cells will fire at particular times and phases of an ongoing brain rhythm in the same area, known as a theta oscillation, which cycles about 8 times per second. When the animal moves faster, these signals are more dominant, and the timing of these cells relative to these phases of the rhythm are thought to be critical for organizing time and space and ultimately contributing to an animal's ability to navigate and remember how to navigate an environment. Now, there are also higher frequency oscillations above 30 hertz that couple to these theta oscillations in a similar way as the cellular activities and may in fact reflect those activities, such that they get stronger at particular phases of an ongoing rhythm. And this theta gambit coupling has been shown also to be critical for this process by many, many people's labs, including Laura Colgan as well. Now, I went to do my Ph.D. at UCLA, and I was privileged to work with Barbara Knowlton and Susan Burkheimer, and collaborated with a neurosurgeon, a pioneering neurosurgeon there named Itzhak Fried. Itzhak Fried and Eric Benke developed an electrode that you see here that would be implanted in patients like Henry. So instead of patients like Henry receiving surgery to remove bilateral medial temporal lobes, that surgery was no longer done, of course, because of the devastating consequences on memory. Instead, these patients would come to the hospital for 1 to 2 weeks at a time and receive fine wired implants throughout their brain in order to record seizures and localize, hopefully, a very small part of the brain that could thereby be treated. These electrodes can record from local activity from these direct brain regions, and over the past many decades, insights from working with these participants have contributed a great amount of knowledge into our field of neuroscience. Now, at UCLA, this electrode, the Benke-Fried electrode, has small little fine microwires about the diameter of a human hair. And these electrodes can pick up extracellular activity from nearby neurons, ultimately allowing you to record from single cells while human participants are completing memory tasks. Many, many insights have been discovered through these recordings, but there remained a challenge as I was doing my PhD during this time, in that the sessions were constrained to a hospital bed. These patients could not walk around. They were tethered to large recording equipment, and these experimental sessions had to be limited to short time windows. But now, what if I told you that some of these patients go on, after their hospital stay, to receive permanent implants in these regions? in the hippocampus. That's actually what happened when I was a postdoctoral fellow with Itzhak at UCLA. In 2013, the FDA approved a device that could be permanently placed, most often in regions like the hippocampus and the medial temporal lobe. This device will record seizure activity and detect the activity and ultimately stimulate to prevent the seizures. But the stimulation is really limited to, overall maybe an hour or two across the day. So these patients are walking around with these devices, recording from the hippocampus throughout their everyday activities, forming memories, recalling memories. This was sort of a eureka moment for me as a postdoc, because I realized that as these people are walking around, they may be willing to participate in research studies where we can really learn a lot about this brain region. And since 2013, the number of participants implanted with this device has really exponentially grown to right now we're seeing over 6,000 individuals who are walking around. You wouldn't even know that they have a device implanted in their hippocampus. Here's an example of 24 participants with electrodes in bilateral hippocampal regions who, many of them are actually very high functioning and have very good seizure control and can come and volunteer for research studies in our laboratory. Here's one participant who's coming in the lab. We can add wearable sensors. We can add, for motion tracking, we can add non-invasive scalp EEG headsets to integrate and synchronize that activity with the intracranial recordings. We can also use virtual reality headsets to simulate the real-world experiences, but with precise experimental control. Now, this is not an easy task to do. It takes a lot of engineering to synchronize all of these sensors. And this work was really led by a lab member of mine, Urish Topalovic, who's giving a poster right after this talk, if you're interested in learning more about the details. These participants can also be equipped with other wearable sensors for heart rate recordings, skin conductance to measure palm sweating, video audio recordings, eye tracking, pupil size changes, indoor and outdoor motion trackers, and the list goes on, because these developments of these sensors are really growing every day that we see. So here's what it looks like. A participant's walking through the lab. We have synchronized hippocampal recordings shown on the bottom right, and we have the ability to track their eyes, the gaze position, as well as their position in the room. All of this activity is synchronized with millisecond resolution, and we can even view it wirelessly as we're standing outside of the room. So what I'd like to talk to you about today is 4 things. I'll spend most of the talk talking to you about how this platform has enabled us to discover some interesting phenomenon related to memory and navigation. But I'll also talk to you about how we're more recently taking these tools out into the world. There are a lot of wearable sensors that are coming into the market that participants can take with them, and we've developed ways to synchronize them with the intracranial activity. I'll also tell you about an exciting new technological platform that allows us to record from single cells as participants are walking around in their hospital room and the hospital corridors. And lastly, I'll tell you how we're using all of this information that we learn to develop new treatments for patients with memory disorders, and I'll focus today on post-traumatic stress disorder. All right, so let's get started. One thing that we wanted to look for first is, do theta oscillations exist in the human hippocampus as people walk around? And in fact, they do. We've replicated this finding across several studies. They exist, but they're not there all the time. They come in intermittent bouts. These bouts last about 2 to 3 cycles of activity. They're short, and they're only there about 10 to 20 to 30% of the time. Now, what's interesting is that the timing of these bouts are critically modulated by the tasks that they're performing. And you may notice that these increases in amplitude of these theta oscillations are happening right before this person is making turns. So what are these bouts modulated by? We wanted to test that through a study led by Martin Sieber in my lab, where he had individuals learn and memorize a certain walking trajectory. This walking trajectory consisted of a right-hand walking trajectory with 4 turns and a left hand. Each individual line shows single trace or single trials from participants who were able to learn this specific path. They had to memorize exactly when and how to turn, and you might notice that one participant added an extra turn here, which actually ended up being very interesting because we could see how this deviation in behavior related to neural activity. When you project theta activity onto the walking path, you notice an interesting pattern, that these bouts are occurring right around these turns, such that they're increasing, shown in the red colors, right before the participants make these turns. Now notice participant 2, who added that extra turn. Even there, you see an increase in these theta activities around that turn. This was not the case for other frequency bands, and Martin confirmed that this was not driven by physical changes in movement speed, eye movements, turning, or head direction, and so on. That it really seemed to be driven by these key moments that were critical for accomplishing these tasks, knowing exactly when and how to turn. Now, you may have noticed a little gap in the middle of this path here because there was a treadmill. And on this treadmill, between trials, participants were asked to walk. Initially, just walk. You can think of anything you want. You can think about what you had for breakfast this morning. But in later trials, Martin asked them to mentally re-experience the walk that they had learned earlier. So during these imagination navigation trials, he could then look at the activity in these electrodes and project them onto the walking path. And what he found was some similarities in the timing and the increases and decreases of these bouts that compared and shared some dynamics with real-world navigation. In fact, if you compared to the control walks on this treadmill, where essentially physical behavior is exactly the same, you see something different. You see that during the imagination trials, there's higher temporal correlation from trial to trial, suggesting that the activity is sort of reactivated or replaying as they imagine these walks. You don't see this in the control condition, and of course you see this much higher in the real-world condition, because imagining experience is a quite variable task. We don't do it so perfectly each time. In fact, we tend to compress these experiences in our minds. Although Martin ensured that people try to take as long as they can and really mimic what they were doing in the real world. Martin also found that the electrodes that showed the highest correlations across trials during real-world walks also showed the highest correlations across trials in the imagined walks, and that these electrodes were located in the hippocampus as opposed to other nearby regions like the entorhinal cortex. Matthias Stangl, a former lab member who now has his own lab at Boston University, wanted to see if these activities could be modulated not by our own walking behavior, but by watching someone else. And in this task, which was also a memory task, it was in a smaller room where the walls formed critical landmarks for how they were to navigate through the room. He found that these activities would increase as they got closer to the walls. So theta activity would increase as they got closer to the walls and decrease as they got further away. And this was true across all of the participants that we tested with significant differences. Now, as you can see in the next video, the participant's actually sitting in the corner of the room and watching Matthias walk around. And they're asked to answer some memory questions, but related to Matthias's position in the room, not their own. And what you're going to see is that theta activity increases not now to when Matthias is close to the walls and decreases as he goes away, suggesting that these activities, the same mechanisms that are activated for one's own position in space, could be similarly modulated by the behavior of another person. This effect was only present when they were engaged in a memory-guided task. So you see this difference between positions in the boundaries versus the inner areas for both self-navigation and observation, but only when they're asked to do a memory task, not when they're asked to visually just walk to a cue in the room, suggesting that these dynamics are really, modulated by this memory-engaged process. He also found that gamma activity showed similar patterns, such that it increased as they got closer to the walls, and that the timing of this gamma activity was locked to particular phases of the ongoing theta, such that it increased at the rising phase, consistent with a lot of findings in rodent studies. If you'd like to learn more about follow-up studies, Matthias's lab has some posters on Tuesday afternoon. Sabrina, Mayo's a graduate student in my lab, wanted to understand whether these activities were modulated by memory performance. So she designed a virtual reality navigation task where they had to learn the location of several colored translucent cylinders that you see here. They also had to walk to some aero control trials where they just simply had to walk to them. There was no requirement for them to remember where they were. They would then go on to learn other locations of different objects, and they would keep doing this until they would learn all of the locations, and then she would have them switch to a different room, let's say a wooden paneled room, where she could then introduce new objects and new positions. A little bit more of a challenging memory task. And what she found is that these theta activities, which I'll show you in the next slide actually, increase during particular moments during retrieval. So here shows an example retrieval trial where they're walking, you can see their path, and they push a button, which is shown with an X. That button is their memory for where this red cylinder was located, and it's pretty good, it's pretty close. They then go on to a control arrow trial, and now in the next trial you're going to see their button press is a little bit further away from the actual position of this green cylinder. So this would be a trial where their memory is not so accurate. And so we had a mix of these trials, some trials where they were doing very well compared to ones where they were not so correct. And what she found was that these theta activities were stronger when they were correct, when they were closer to the actual position of these objects. She also didn't see that these activities were driven during the visually cued navigation trials, similar to our previous studies. And she also found that when she dynamically switched the context, which you can do nicely in virtual reality, that these activity patterns would remap, such that they would be specific to the locations and the objects in the other room. Overall, this suggests that these activities are dynamic, and they're also sensitive to the success of the memory being recalled. This just shows you the timing of that effect, such that it's increasing prior to their arrival at the target, and not for the other conditions, such as incorrect trials or visible or arrow trials. She also confirmed that this wasn't driven by other variables, such as movement speed, angular velocity, and head direction, and it really was memory performance that explained the most amount of variance in these theta activities. Now, Corey Inman in the lab, a former lab member who now has his own lab at University of Utah, did something really kind of wild, which is he took these recordings out into the real world. UCLA, if you've ever been to UCLA, south side of campus, is a very confusing area of campus. I spent close to 20 years of my life there and probably would still get lost. So we had participants walk around a very convoluted route on the south side of UCLA campus and had them learn this route and complete it repeatedly over the course of the day. In fact, he recorded a staggering 28.5 miles of walking in 5 participants who had these hippocampal and medial temporal electrodes. He also measured their eye movements and movement speeds and other variables, and found that amongst these experiences, there were many moments where the participants would just naturally get lost. And you could see this often in their verbal reports. They would hesitate, they would start looking around, so their fixations would go down, and their velocity would decrease. They may even have sort of a delayed sweating response on their palms because they're nervous that the experimenters are watching them and seeing them mess up. What he found in terms of the theta activities is that there was an increase and then a major drop in activity once they were able to find their way. And I'm going to show you a video of this, but you can see here this decrease, or the blue colors illustrating this significant drop in theta activity. So here's one participant where they're walking. You can see what they see, and the green circle is signifying their gaze position. They're supposed to make a right turn at this upcoming fork in the road. You can see increases in lower frequency activity prior, where they're sort of confused and hesitating.
He did look kind of familiar.
I said, okay, this does kind of look familiar, and they go along their way, at which point you see this major drop in theta and low frequency activity, suggesting that during this sort of period of eureka or finding their way, that this system is sort of shutting down and not participating this in sort of internal generation comparison to the external world process. Now, what you may have noticed is that speed is actually increasing once they find their way. It decreases at first when they're lost, and then it increases. But theta is going down. And we know from rodent studies that theta has a very prominent relationship with movement speed. So I want to dig into that a little bit further to tell you that in all of the memory tasks that we've run, we don't see this relationship with movement speed. So speed doesn't explain the changes in these theta activities. However, when you look at the non-memory guided navigation tasks, you do see a relationship with movement speed, such that when they're walking faster, you see increases in the prevalence of these bouts versus when they're walking slower, suggesting that walking speed relationships with theta are present and consistent with rodent studies, but only when memory is not engaged. Now, what about eye movements? We do see some significant explanation of theta activities relating to eye movements. So let's dig into this a little bit further. Hamza Zubair, a graduate student in the lab, looked at some of our existing data sets and found that theta does in fact increase during periods where there are more saccades. But this is only the case during the memory-guided navigation tasks. He also found that it increases during saccadic behaviors that have sort of higher chaos and more exploratory patterns, and also when they're making longer distance saccades compared to shorter ones. And consistent with non-human primate visual studies, he sees a resetting in the phase of theta as saccades are occurring. So let's summarize at this point everything we've discussed so far. We see that hippocampal theta activity comes in bouts, and that the timing of these bouts are critically modulated by the task that people are doing. They're highly engaged during memory-guided navigation tasks, but they could also be internally generated through these imagined navigation processes. They're also stronger during successful versus unsuccessful memory. They are linked to saccadic eye movements when memory is engaged. When memory is not engaged, they can show relationships to movement speed. They are modulated by the position of others in addition to one's own behavior, and they are involved in sort of this getting lost behavior, but are shutting down during this process of finding our way. And also mention gamma shows up a lot in here, and reiterate that it is highly coupled to these theta activities. So to summarize, we think that what's happening here is that these signals are reflecting a sort of process of internal and external navigation, where we're essentially using this two pieces of information to update our model or our understanding of the world around us and our memories. Okay, so we've spent the larger part of the last decade on the studies that I've shared to you today, but more recently, we've been able to start to move these experiments out into the real world. So how are we doing this? Well, it turns out you may have seen on the market the emergence of these wearable devices that can capture first-person experience. And we have used such a device, such as the Meta Ray-Bans, to integrate this with a platform that allows us to synchronize the implants with the first-person video. This is a development through Uroš Topalovic and Nick Haselak. And what they've built here is the camera-embedded glasses. They've built an LED that they put on the frame that is connected to a wearable head-mounted electromagnet. This electromagnet could introduce signals via a manual button press into the implanted device to trigger the storage of 2 minutes behind, 1 minute ahead, and essentially allow you to have a signal in the intracranial data that is synchronized to the LED that's captured in the first-person video. We've employed this platform in 5 participants so far who've captured a wide, diverse array of experiences, such as cooking, hanging out at the beach, gardening, visiting the farmer's market, walking their dogs, everyday activities, and memories that are forming. Wenshin has captured, in 5 participants, over 130 of these episodes. Here's an example of one such episode where a participant is visiting their local Chinatown while synchronized hippocampal and parietal activities recorded. Now, often when we have these events that we capture, we will replay them on our phone. In fact, I have a 3-year-old who really is obsessed with looking at all of these old videos of her birthday and other events. We do this all the time. So what if we measured activity during this process? We could replay these videos via an app called Hippocamera that was developed by Morgan Brentz's lab, and allows us to really control which videos are played. Some videos are played more often than others, and here you see an example of how these replays look. This participant is viewing an event of watching their mother cook, and overall, in 5 participants, Wenshin has captured over 1,800 of these memory replays over several months over the course of 3 to 4 months. Here's the memory that I just showed you of them visiting their local Chinatown. And we can start to look at how the activity during these memory replays evolves over time, where some memories are replayed more heavily than others. And I won't tell you the results because you'll have to go to Wenshin's talk on Tuesday, but there's some very exciting preliminary results suggesting that over time we may start to be able to decode or separate these memories based on the time course of activities, especially theta activity in the hippocampus. I'd like to now tell you a little bit about our new methods for recording from single neurons while participants are walking and navigating and participating in memory tasks. And this is really possible due to really technological advancements with miniaturization of the electronics that record from these electrodes. One such device shown here is called the Neurostack, which was developed through collaborators of ours at UCLA, Dan Markovich. And you can see examples of 12 channels of recordings from the microwires, from the Benke-Fried microwires, that are enabling us to see single spikes or extracellular spikes from nearby neurons. We could then use clustering algorithms to assign these spikes to different units and essentially record single unit activities while participants are walking around, synchronized with motion tracking, eye tracking, and other devices. Now, the exciting aspect of this device, through developments and funding from the BRAIN Initiative, is that the electronics have actually been miniaturized to fit within a single pill-sized device. This opportunity presents an enormous, sort of exciting potential for the future, whereby participants such as these epilepsy patients like HM who now get electrodes implanted could essentially, be going home with an implanted device that can record. Each device can record from 32 channels of single units, microwires, and up to 128 LFP. And together can really formulate a sort of, outstanding number of recordings. So we've used this device and other sort of similar devices that have come onto the market, such as from Ripple. And we've recorded from single units. As a participant example here, you could see is walking around in their hospital room. So on the bottom, you see the little white dots signifying these spikes from different channels of microwires. And as they're walking around, there's an augmented reality headset that can show them stimuli, such as a red square on the screen or other images. And they are asked to learn different mappings of images to different parts of the room. When they see a red square, they're asked to recall images, a certain mapping of these images to particular parts of the corridor. Then we can change the context using these blue cues and essentially study how navigation and memory are intertwined and affect these single unit recordings. Now, Wenshin is also going to talk about this in her Tuesday talk, but I'll sort of preface to say that there is a mix of exciting responses from these units, such as cells that will increase when the person is in particular parts of the Y maze, as well as of responses that are happening prior to their verbal recall of these image locations. Lastly, I'd like to tell you about how we're using all of this information to generate new therapies for patients with memory disorders, and I'll focus today on post-traumatic stress disorder. But before I tell you about that, I want to go back in history a little bit. So over a half a century ago, a neurosurgeon named Wilder Penfield would stimulate individuals' brains as they were on the on the operating table, in order to map out different regions such that they would ideally prevent the removal of a critical area. And sometimes when they were stimulating the temporal lobe, participants or patients would have these very, very vivid recollections from their past. In fact, one patient suddenly saw herself in childbirth, having had 2 children. This sounds quite horrifying to me, reliving the experience. In fact, these patients report that these experiences, when triggered electrically, seem complete, including all of the things that the individual was aware of at the time. More recent reports from patients with epilepsy from Josh Jacobs' lab have shown that these memories or these can be triggered by stimulation, again, from the temporal lobe, such as this patient remembering stuff from high school. And if you look at all of these reports, these observations over the past many, many decades, you see a pattern such that amygdala and hippocampal stimulation seems to most often trigger these very specific episodic or personal memories. And the vividness of these memories seems to get stronger as the stimulation strength increases. So we wondered whether there would be some sort of electrical signature or excessive activity may be related to theta and gamma in these regions that give rise to the flashbacks in PTSD. And if this was the case, could we then use those signals to trigger a therapy to stimulate and prevent these episodes? Now, this approach is not new. It's actually being used in epilepsy, which I'll tell you about. But before I share that with you, I want to share with you a little video from our first patient in the clinical trial. This is Sergeant Adam Holbrook. He's a combat veteran who suffered from treatment-resistant post-traumatic stress disorder. And he's going to tell you what one of these flashbacks are like. Because if you've never experienced them, it's actually very difficult to imagine really how vivid these experiences are.
And all of a sudden it just hit hard with a ton of bricks where I started seeing things that weren't there. I thought it was just all normal stuff coming back to me, like visuals and memories and everything, but it got real intense. Be on like a patrol all of a sudden, and I'm not on a patrol, I'm in Target. I've had hospital visits, I've had times where I've been blacked out on the ground, and ambulance care guys are just sitting there like trying to figure out what's wrong with me. They don't know what's wrong with me. I couldn't express what it was going on. I just knew it was like about war.
So as you heard, these emotional events, the intensity and the sensory detail of these moments are quite striking. In fact, mirroring in some ways the vivid immersive qualities of the electrically induced memories described by Penfield and others. So we, again, wondered whether there was some sort of change electrically in these regions that would give rise to these flashbacks, and we wanted to see if we could, record and detect them. So we underwent a clinical trial through collaboration with J.P. Langevin and Ralph Koch at UCLA, where 6 participants with treatment-resistant PTSD were implanted with these same devices that were used in epilepsy in the hippocampus and amygdala region. Now, participants could swipe when they would have these flashbacks. If they could swipe, they would capture these events and allow us to see what's happening. And we took all of these events, not just the flashbacks, but any kind of negative symptoms that they were having, and compared the activity in these regions for those events compared to neutral periods that we had programmatically recorded. And what Mauricio Vallejo found, who led this analysis, that theta power and as well as gamma increased during these negative symptoms compared to these neutral periods, suggesting that there may in fact be a signal that we could help us detect these instances from others. Another study that provides some evidence for a potential signal that we can detect comes from Anthony Yang, who is giving a talk on Wednesday morning. And what he did is he had participants come into the lab and go through basically a fear conditioning experiment. Now, this task allowed participants to learn associations between colored lights or neutral cues and aversive outcomes, such as a spider jumping on their face. And what he found is that hippocampal and amygdala activity, particularly theta activity, increased during the cue that was associated with the aversive outcome, the green light, as opposed to another light that was never associated. I'd like to show you a video of this because it's really quite fun and interesting to see. What you're going to see is what the participant sees on the top left right, which is a virtual reality environment. It's a grocery store. We have many of these that are developed by our lab artist, Emmy Jenkins. And as they're walking through, there may be a light color change. And right after that, there will be a spider that jumps on the screen. So I just want to warn you that it might be a little bit startling. You'll see also recorded the intracranial EEG, the pulse, the skin conductance or sweating on the palms, and their position in the room. On the bottom left, you can see the participant walking through the room, and as they're walking, again, a light color change happens. Sorry about that. So when that spider jumps on the headset, and there's a little electrode that kind of vibrates the headset, it's quite aversive, and you see an increase in skin conductance that you can see right over here, and an increase in theta activity. And over time, as they learn this association, that increase starts to happen earlier to the actual light. So this is a blue light. There's nothing that's going to happen, so you don't have to worry. And we can start to see the difference in activity patterns, both in the brain and in behavior to these different cues. All right, these 2 studies suggest to us that there is a signal that we may be able to detect with the commercially available FDA-approved device that's used for epilepsy. This is the NeuroPace RNS, which again is designed to record seizure activity and then stimulate in order to prevent the seizures. But in this case, we're going to use low-frequency activity to trigger the stimulation, and the stimulation will be to this amygdala hippocampal region. We've already reported on our first 2 participants, but what I'm going to show you is data from our 5 out of our 6 participants that have completed the study so far. So the clinical results are quite exciting and striking. On the y-axis, you can see the clinician-administered PTSD scale, which is how we measure symptoms in PTSD. The lower the scores, the better the patient is doing. In fact, many of these patients have reached the threshold of remission, some very quickly within the first few months, and Adam, in fact, who you can see in blue, reached remission and stayed in remission for over 4 years. Now, one patient is improving, but not quite as significant as the others, and we recently noticed that the electrode placement is a little bit more posterior than the others, and also this participant's data doesn't show a relationship with a sort of change in the excitatory-inhibitory balance of these regions. So that's what this graph shows here. On the y-axis, you can see their CAP scores. Remember, lower the scores, the better they're doing. And what we see in the responders is that as they improve, the sort of balance between high frequency and low frequency activity changes, such that the gamma activity essentially is going down over time, especially when they start to improve, and that's not happening in this non-responder. This suggests to us, again, that there is some sort of mechanism here of response that we can explore further and help us understand why this works for some and not others. So these are very exciting to us. We're hoping to expand and really try to understand what's going on. But I do want to share with you, in Adam's own words, what it's been like with him to participate, what it's been like for him to participate in this trial.
All in all, the experience has been wonderful. I came in with little hope, because it's just the way that things were going in my life, and we put the device in, it got turned on, and then like a couple days later I started getting these funny little like tingling sensations throughout the body. It's the washing over feeling, like you've been run over by water. I had been triggered pretty severely actually at that time, and it all of a sudden just like cloaked over me, and I was fine.
This experience that he's sharing with you actually was Disneyland, being able to watch the fireworks for the first time with his family. It's just an enormous change. He would never have been able to experience or watch the fireworks, given that these are very loud sounds that can trigger his flashbacks. We are excited to say that Adam's been in remission for over 4 years, and he hasn't had a single dissociative flashback since this therapy was turned on. All right, so we're excited for these preliminary results. We're hoping to understand further how it's all working, but I'll sort of summarize here to say that we've developed these tools in the lab to start to be able to understand these brain regions during more naturalistic behaviors, freely moving behaviors, and then moving this out into the real world and recording from single neurons and ultimately developing and testing new therapies. So it's a very exciting time for our lab to be able to build on this first decade of our work. But since we're only interested in memory, I really just want to emphasize that these tools can be applied to the study of various other cognitive functions in neuroscience, and I hope that others will sort of pick up these tools to apply them in other ways. We have already started to share these tools with other labs who are developing clinical trials for other disorders, such as obsessive-compulsive disorder. In these studies, the electrode site varies. However, the approach is the same, which is that they will record from these devices during naturalistic behaviors, for instance, compulsions in OCD, and then develop closed-loop therapies using these devices to prevent symptoms, and have had some promising results so far. We're also collaborating with individuals who are looking at Parkinson's disease and hoping to detect freezing episodes and prevent these symptoms in those patients. And lastly, we've successfully collaborated with Casey Halpern's lab to study loss of controlled eating and prevent these compulsive behaviors in binge eating disorder. So I'd like to end the talk by highlighting a larger initiative that goes beyond my lab that we are very grateful to be funded and supported by. And these are initiatives that are moving neuroscience into more naturalistic spaces. This is the Simons collaboration on ecological neuroscience, and if you're interested in some of the groups that are working on this, there will be a symposium on Tuesday afternoon organized by Nachum Alanovsky. The Brain Initiative has also been huge for moving us into a space where we have the tools to capture this very complex multimodal behavior. And so this program, the BBQS, is really pushing forward the development of these sensors, not just for the brain, but for the body, and developing tools that allows us to analyze the complexity of these complex data sets, such as using AI and machine learning techniques. So if you're interested in that, you can see some posters and scan the QR code here. Most importantly, I'd like to thank all of the patients and participants who volunteered for this work. This story really does start with a patient, and that's Henry Molaison, and none of this work would be possible without the time and commitment of these patients. So we're very grateful to work with these individuals. They've really become sort of lab members to us. And also all of the many clinicians who've worked with us, the surgeons, the neurologists, the psychiatrists, the engineers. This really takes a village to do these kinds of studies. Last but not least, I'd like to thank my lab members. It's been an incredible journey working with these individuals. I've highlighted several of their studies today, and many of them are actually here at SFN giving their own posters and their own talks. These brilliant people have really made this journey beyond words that I can describe, and I'm so grateful to work with them. And lastly, just to bring us to here today, we have moved the lab to Duke University. Most of the entire lab has moved with us across country, and I'm very grateful to Duke for them creating this dream space for us that we can really embark on the next, hopefully several decades of studies and discoveries. And if you're interested in any of the work that will be presented at SFN, you can scan a QR code there to see the schedule of events. So with that, thank you very much, and I'm happy to take questions.